Hi, my name is Nur Haslina binti Muhammad Sultan. In this video, I will explain about microalgae culture, counting microalgae, rotifer culture, and microworm culture. Microalgae culture. These are the materials needed. Microalgae are microscopic algae and typically found in both freshwater and marine systems. It is suitable as food for larvae because it have appropriate cell size for ingestion, rapid growth rates, and contain good nutrient composition. After 10 days, color intensity of the culture increase along the culture period indicates that there are cell growth where chlorophyll pigment increase. Aeration or mixing the algae one time per week is very important to avoid sedimentation of the algae and to ensure all cells are equally exposed to the light. Counting microalgae. These are the materials required. First, insert the sample using larger pipette and put it into test tube. Then insert 1 drop of Lugo solution that we made before into the sample test tube. Hence take the Neubauer hemocytometer, put the slides on top of it, then pipetting the solution into it. As for the result, observe it under light microscope with low magnification. Counting algae started with the center box. Do count those on the top and right lines such as this. Do not count those on the bottom and left lines such those. We do this to avoid counting the same cell multiple times. Rotifer culture. Materials required are: first of all, the rigid tubing must be attached to the flexible tubing and to the air pump. Smooth airflow is important to avoid female rotifers to lose their eggs. Add a few ounces of phytoplankton into a starter rotifer culture. When the culture turns into light green tea color, that means the rotifers has fed. It's time to start the second culture. Take 1/3 of the culture and pour it into a second plastic bottle. To filter, pour about 3/4 of the culture into a rotifer sieve and into a discard bucket. The concentrated rotifers were collected. Feed them to the larvae or place them in a refrigerator for the future use. Microworm culture. The materials needed are: add 1-inch layer of cooked oatmeal to the bottom of the container. Make sure to poke some holes on the lid to let air in. If not, the culture will die. Let the oatmeal cool overnight and the active dry yeast were added. The microworm culture was added to the cooked oatmeal. Stir it once a week to keep it alive. Using Q-tips, swab the small amount of microworm on the side of the container. And using dropper, the microworm was taken from the chlorine-free water, inject the microworm into the fry tank. Culturing live food is important for larval nutrition due to their very small size for increasing survival rate, feeding efficiency, and growth rates. The suitability of feed use are the major factor determining the profitability of aquaculture operation.